Hi, my name is Anika, and I took AP Bio last year. I did get a 5, so you're in good hands. And today I'm pretty much just going to be going over cells with you and just some basic information. I'm not going to get too in-depth, just as a kind of refresher. So some basic information that you need to know. So if you guys want to drop in the chat how you're feeling about cells or about AP Bio in general, because since the year just started, see how you're liking it. Let's see, a scale from 1 to 10. How are you feeling about bio so far? Don't be scared. Okay, well, while you guys are typing, I'm going to go ahead and start. We are going to have a question and answer at the end of this, so if you guys come up with any questions, please send them. So make sure you follow Fiveable on all of our social media, Twitter, Instagram, YouTube, at Think Fiveable, so you can get updates on any AP class that you have, not just bio. And so you can watch other streams, not just biology or just me, other streams that you have going on to help you with your classes. Okay, so today in the stream, I'm going to be going over some basic information about cells, like prokaryotes versus eukaryotes, and the things that make them different and the things that they have in common. Cell structure, I'm going to be going over what the cell membrane, its structure and function, and the different organelles of the cell. Pretty basic information, though. I'm not going to be getting too in-depth with the different organelles. I'm also going to be going over the different types of cellular transport that there are. And at the end, I'm going to be doing a question and answer. So if you have any questions that aren't like burning, that need to be answered right away, you can go ahead and put it in the ask a question box, and I will get to it at the end. But if it is something that's burning and you need like an answer as soon as possible right away, you can put it in the chat and I will answer it as soon as I can. Okay, so we are going to go ahead and start. Hopefully more people will be joining us. So prokaryotes versus eukaryotes. So prokaryotes are simple. They have no nucleus, no organelles, and they're single-celled. So pretty much a prokaryote, an example would be a lot of types of bacteria are prokaryotes. So they're pretty simple compared to like us as eukaryotes. If you want to compare us to a bacteria, there are a lot of differences and we're very complex in comparison. So eukaryotes are a lot more complex and our cells have a nucleus. They also have organelles, which are different parts of the cell that perform a lot of different functions. And they also have, they also can be single-celled or multicellular. So eukaryotes, like we're a eukaryote, but also other types of bacteria are also eukaryotes. So it kind of varies. But in general, we are definitely more complex, eukaryotes. Both of them have genetic material because after all, you do need some genes in order to function, in order to live. You can't really live without some form of genetic material, be it DNA or RNA. And they also both have a cell membrane to pretty much keep the cell in one place, because if there was no cell membrane, the cell would just be all over the place. The contents of the cell would spill out. Cytoplasm, again, to kind of hold things in place. Ribosomes to produce proteins, because actually what I found interesting to learn when I was in bio is that proteins are what we're made of. Like nothing is made without proteins. Proteins are how DNA expresses itself. So just like you need DNA, you also need proteins. The word eukaryotes, I think it does come from Latin. I wouldn't be surprised because a lot of biology terms do come from Latin, but I'm not actually sure. So if you find out, answer it and let me know. But I do think it is Latin, but I don't know for sure. Good question. That's interesting. So also both of them can have a cell wall. So you have, although not all eukaryotes have a cell wall, we don't have cell walls. That's only in plants. We can. Eukaryotes covers plants and animals, so it can have a cell wall. Okay, so we're just going to go over some of the things that prokaryotes have specifically that eukaryotes don't have, and then we'll get into the things that eukaryotes have. So pilus, so this is an image of a prokaryote. I think it's some sort of bacteria, but it's not like anyone in specific. Pilus is used to adhere to other cells. It's kind of cool. It's also used to some bacteria can it's something called conjugation, where they almost like transfer some of their DNA to another bacteria. So pili is the plural form of the word, and they're used to attach to the other cells and transfer the DNA. I think that's really cool. Imagine if we could do that. That'd be really awesome. Okay, capsule is additional protection for the prokaryotic cell. So like bacteria, they exist in like a lot harsher environments than we tend to exist in. So like bacteria can exist in like, for example, bacteria enter our bodies to like infect us. So they need extra protection on top of like what they usually, on top of like regular cell protection. So that's pretty much what the capsule is. Nucleoid is the DNA region. So like they don't have a set nucleus. It's not like enclosed in a membrane, but pretty much nucleoid is just their DNA super condensed that all tends to kind of stay together. Flagellum is used for movement. So it pretty much like kind of is like a tail that propels them throughout whatever substance they're in, so like water. And actually, flagellum is not only found in prokaryotes, but it usually is. So if you see something with a flagellum, chances are it's a prokaryote, but not 100% of the time. For example, human sperm cells have flagellum. They're one of the eukaryotic cells with it. So here, now we have a practice question. So studying a picture of a cell taken with an electron microscope, you find that the cell has no nucleus and no mitochondria, but it does have a plasma membrane and a cell wall. You conclude that the cell is probably from a. So if you guys want to put your answers in the chat, and I'll give you guys like a minute or so to think about it, and I will also go through my thought process after you guys come up with some answers. Please send answers, don't be scared. It's okay. If you're wrong, then that's helpful because I can help figure out why you had that misconception. So I'll give you guys a little bit longer. Okay, so since no one is answering, I'm going to start going through. Thank you, Zoe. It's not, okay, so the answer is C, and I'll go through my thought process here. So it's definitely not D, right? Because extinct organism is like completely like irrelevant. Like that's just like a distractor choice, you know? So you can eliminate D. Like that is unrelated to the fact of these structures being present, right? And then you have animal, plant, and prokaryote left. So the fact that there's no nucleus and no mitochondria, that means it cannot be eukaryotic at all. Like all plants and all animals have a nucleus and they all have mitochondria. You need mitochondria to produce energy and you need your nucleus to house your DNA. So you know that it can't be animal or plant because those both fall under eukaryotes and all eukaryotes have nucleus and mitochondria. And so it is prokaryote. The answer is C for those of you who put C in the chat. Prokaryotes do have a plasma membrane. Everything has a plasma membrane, so that doesn't really help you very much. But what they do give you is that it also has a cell wall. So from that, if you were to look at it that way instead of the way I just looked at it, you could also narrow it down to either plant or prokaryote. And then when you see there's no nucleus and no mitochondria, then you can choose prokaryote. So that's how I answer most of my questions, actually, multiple choice wise in AP Bio. I go from a process of elimination standpoint because I think that's the best way to get like a for sure answer, because if you know it's not any of the other answers, then it must be your answer. So we can go to the next. Okay, so now we're going to go to talking about the cell membrane. So all cells have a cell membrane, whether it's prokaryotic or eukaryotic. Bacteria have cell membranes, we have cell membranes, plants have cell membranes, everything has a cell membrane. So pretty much the model that we use for the fluid mosaic is called the fluid mosaic model. So what do you guys, so in terms of the word mosaic, what does anyone know what a mosaic is? It's a type of art. I was looking it up earlier. It's very pretty. If anyone wants to try to describe what it is, because it helps to remember this. Yes, Zoe, it's like art made of different tiles, and it's like almost like a mural with a whole bunch of different tiles put together. Like when you think of stained glass, like stained glass windows, like in like churches and stuff, it's almost like that, like with a bunch of different tiles pieced together. So pretty much that helps me remember fluid mosaic model, because the word fluid is kind of self-explanatory. It just means that it's constantly moving and it's not like rigid. But mosaic means that there's a whole bunch of different pieces. So pretty much there's a whole bunch of different pieces that are involved in the cell membrane. A whole bunch of different pieces that are involved in the cell membrane, and those make up the cell membrane and the fluid mosaic model because they're constantly moving. Phospholipid bilayer. Okay, so the cell wall is made up of a phospholipid bilayer. Pretty much phospholipids have a phosphate head. So the circle part, if you look at the diagram like really closely, the circle part is the phosphate head and it has a lipid tail. The heads are hydrophilic, which means they like water, because phosphate is polar, which means it likes water, but you don't have to get too deep into the polarity. And then hydrophobic tails are the lipid tails. The way that helps me remember is that lipids are hydrophobic because fat is lipids and like oil. So when you think of trying to mix water and oil, or even like fat, when you're like cooking and the fat always rises to the top because it doesn't like dissolve, it's hydrophobic. So you know that it's phospholipid, tells you that it's phosphate and it's lipid, and you know that one part is the head and one part is the tail. But to help me remember, I remember that the tail is hydrophobic. So then the lipid, which is hydrophobic, makes up the tail. That's just something that helps me to remember. If you guys have anything else that like any hints or tips or tricks to help you remember, feel free to put them in the chat so you can help other people also. So the hydrophobic tails kind of go together in the middle, as you can see in the diagram, and the heads form the outside. So the heads form the outside because the cell is in what's called an extracellular matrix, and it's pretty much just like the fluid that's like between your cells. So that has to be hydrophilic because otherwise it wouldn't really be able to be in there. They'd all like float to the top, which wouldn't work out very well for you. So then the cell membrane is semi-permeable, which pretty much just means that some things can pass while some things can't. So for example, large molecules have a lot of trouble passing through the cell membrane just because they're so big. And also hydrophilic things, which tend to be polar, have trouble passing through, so they need some help, which we'll get to in transport actually. But it's semi-permeable, so it can control what enters and what leaves. They do have a lot of proteins embedded, which is part of what makes it a fluid mosaic, right? Because there's a lot of different things involved. So the proteins embedded in the cell membrane, you have transport proteins, which help substances to enter and exit the cell. So some of them are like, as you can see in the diagram, you have some that are like channels. Those are transport proteins, and you also have some other ones, which we'll talk about when you get to transport. But pretty much transport proteins help substances enter and exit the cell. And then you have receptor proteins, which trigger different actions inside the cell through pretty much cell signaling. So the receptor proteins are located on the outside of the cell, and there'll be some sort of signal that will pretty much trigger the receptor protein, which signals something inside the cell to do some action or whatever it's trying to signal. So that's transport receptor proteins. Then you have structural proteins, which is kind of self-explanatory. They just support. You also have glycoproteins, which are really interesting to me. They help with cell identification. So what helps me remember glycoproteins is the fact that they're involved in determining blood types. So people with type, not the plus and minus, but like type A, B, or O. So people with type A blood have A proteins. People with type B blood have B proteins. People with type AB have both, and people with O have neither. You don't have to know all of this, but it just helped me to remember. It's just something interesting, and I remember interesting things a lot easier. So, for example, people with type A can only take type A blood because their protein, that's how their blood cells recognize each other. If they have some sort of other protein it won't recognize, and your body will reject the blood. So people with type AB blood can donate to people with type A or type B because they have both of these proteins. And people with type O blood have neither. So they can go to anyone because they're not going to be rejected based on having the wrong protein. So glycoproteins pretty much help with cell identification. It's kind of like something that says, hey, I'm a X, I'm a liver cell, I'm a blood cell, and I have protein A. It just kind of helps with cell identification, which in turn helps with different cell communication mechanisms. So we have another practice question. So which of the following best characterizes the structure of the plasma membrane? You have A, rigid and unchanging, B, rigid but varying between cells, C, fluid and disorganized, or D, fluid and organized. So I'll let you guys put your answers in the chat, and then again, I'll do the same thing and go through my thought process with the question, and yeah, so I'll give you guys a second. Okay, so yes, Madeline, it is D. So my thought process on this one is because of the fluid mosaic model, which we just talked about. Pretty much fluid is in the name, so you know that it's not rigid, so that can narrow you down to C and D. And then while it does have a lot of different things going on, it is organized. Like there are places for each thing to be, and each thing has a role, each element, like the different types of proteins. So it's fluid and organized. So I would have narrowed it down to C and D, and then just from there, I know that it can't be disorganized. That just wouldn't logically allow you to function. So then I would have narrowed it down to D, which is the answer. If you guys have any questions, please ask them. And also you can use the ask a question box so we can have answers for the question and answer at the end. Okay, so here's an overview of organelles. I'm going to be going through all of them, but I'm pretty much just going to be talking for a lot of this, so just so you know. So we have, on the left, we have a plant cell, and on the right, we have an animal cell. So plant cells tend to have cell walls, which are really, they help to give the plant cell structure because plants need a little bit more structure than we need. So first, before I get into that, though, let's go over what's common to both of the cells. So you have cytoplasm, which is in all cells, be it prokaryotic, eukaryotic, plant, animal, bacteria, whatever it is. All cells have cytoplasm because cytoplasm is pretty much just the fluid inside of the cell that holds everything in, keeps everything together. So cytoplasm is just what everything is in. It's just like a fluid. There's not much you need to know in terms of the cytoplasm. Both cells also have a nucleus. The nucleus holds DNA in the form of chromatin. So chromatin is pretty much your uncondensed DNA. So the way that my teacher explained it to me, and it helps me a lot, is think of it as sand. Chromatin is like just sand on the floor. But then when you condense it, you can actually see the sand. Like the chromatin is just super uncondensed, like flowy, not really rigid or anything. So that's what's in the nucleus for most of the cell's lifespan. It holds DNA in the form of chromatin. The nucleus also has a nuclear envelope that kind of keeps all the DNA inside the nucleus. It's also made of a phospholipid bilayer. So the nucleus has this nuclear envelope and it's also selectively permeable. Some things can get in, some things can get out, can't get in, and it holds the DNA in the form of chromatin. And then you have mitochondria. So I'm pretty sure all of you probably have heard mitochondria is the powerhouse of the cell. But that doesn't really help you very much when you're thinking about it. So mitochondria produces energy. So then if you want to think of powerhouse, energy, power, that if you want to think of it that way. So it produces energy through cellular respiration, which we're not going to get too into, but pretty much it's just the way you produce energy in the form of ATP. ATP is cellular energy. So then you have the lysosome, which I think is really cool. Lysosome pretty much digests old cell parts and microorganisms that can be inside the cell. So they're kind of like a garbage truck. They go and they digest. They have different digestive enzymes inside of them, so they can go and like engulf other things and then like digest them. Like if the cell has something that it doesn't need anymore or something like that, then the lysosome comes and digests it, which I think is really cool. You also have the Golgi apparatus, which is also called the Golgi complex. They're the same thing, which packages proteins to be exported outside of the cell. So the Golgi complex packages proteins that come from the endoplasmic reticulum, which we'll get into in a second, but it pretty much just packages them and sends them off in vesicles. So the endoplasmic reticulum here, you have rough and smooth. So starting with smooth, the smooth endoplasmic reticulum is smooth because it doesn't have like ribosomes on it. It's smooth. And it produces hormones for the body, and it also detoxes the body. So those are the two main functions in terms of the smooth endoplasmic reticulum: hormone production and detoxification. But what you usually are asked a little bit more about, and you'll probably need to know more about, would be the rough endoplasmic reticulum. So it's called rough because it has ribosomes like studded on it. So that's why it's called rough, and it helps you to remember what it does. The rough endoplasmic reticulum produces proteins, and these proteins produced here are used to be exported out of the cell. So there's like a certain order in which proteins to be exported out of the cell go through, and I've noticed that I've seen like a lot of questions on that. Like last year when I took the exam, in all of my practice tests, and even on the actual exam, I feel like everywhere I go there are questions on that. So the rough so the path that a protein to be exported out of the cell takes is it goes from rough endoplasmic reticulum to the Golgi apparatus. So it's produced in the rough endoplasmic reticulum. Then the Golgi packages it and sends it off in a vesicle, and the vesicle fuses with the cell membrane and sends it outside. I have a picture in Transport that really explains this, and I'll keep mentioning it just because I feel like it's really important. So yeah. And then you have ribosomes. Ribosomes are just protein factories. They produce proteins. That's really it. you learn more about protein production later in the year, but in terms of just generally the function of ribosomes is to produce proteins. Ribosomes can be free or on the endoplasmic reticulum. So as I said just a few seconds ago, ribosomes on the endoplasmic reticulum are to be exported out of the cell. So they are the ones that are to be sent to another cell or somewhere else in the body, somewhere else in the organism. Whereas ribosomes that are free, so ribosomes that are just floating in the cytoplasm, as you saw in the prokaryotic cell, I'm going to go back really quick. Here, the prokaryotic cell, those little dots, those are ribosomes. They're also in eukaryotic cells, but they're kind of hard to see in the picture that I chose. But the dots are ribosomes, so they're just free-floating. They can be free-floating or attached to the endoplasmic reticulum. The ones that are free-floating are used to produce proteins that are going to be actually used inside of the cell. So they're used to produce proteins that are not going to be exported, pretty much. And then vesicles are just transport mechanisms for the cell. They pretty much just hold things to be transported into or out of the cell. Yeah, they're just like cars. I like to think of a cell in terms of a city, which I think I have a slide on that coming up, but it helps me to remember things a lot better. So just quick thing on plant versus animal cells. So plant cells have a cell wall, central vacuole, and chloroplast, whereas animal cells have a centrosome. So these are the two things that are like unique to each type of cell. So plant cells, they have a cell wall, which pretty much just gives it a little bit of extra structure, extra support. Animal cells do not. Plant cells also have what's called a central vacuole, which is used for water storage. And some interesting information about the central vacuole, which helps me to remember like its function, is the fact that when you guys have seen a plant wilted, right? So when a plant is wilted, it's because it doesn't have enough water in the central vacuole. So the central vacuole kind of fills it up and gives it makes it firm, I guess. So when the central vacuole is empty or it's not getting enough water, it gets wilted and kind of not firm anymore. So that helps me to remember. Cool facts like that help me to remember things. So that's a central vacuole. And then chloroplasts are used for photosynthesis in plants. So that one's kind of easy to remember. It just happens does not happen in animals because we don't conduct photosynthesis. Animal cells have a centrosome. So that diagram there, you don't really need to know like the different parts of the centrosome, but just generally that animal cells have the centrosome. Centrosome is the same exact thing as two centrioles. So like those two centrioles that look kind of like noodles, kind of like pasta, they make up one centrosome. So if the two words are like interchangeable, so don't get confused if you see both of them. But it's used for cell division. So in cell division, they form something called a spindle, which helps the cell to divide, which plant cells don't need because they have a cell wall. Okay, so as I was saying a little bit ago about the cell as a city, this is what helps me to remember the cell, just because I really like mnemonics. So the cell is a city. So you have the cell membrane, which is just like the boundaries of the city limits. You also have, and that's what the cell membrane is in a cell, pretty much just the boundary. The cell wall in plants is just like fortification. So I think of it as like an actual wall, or like in like those ancient structures when you have like a wall and like a moat and all of that. That's what the cell wall is pretty much. It's like extra support. Nucleus is City Hall because it houses everyone's information, all of the information about like everything, which is your DNA. So it has like all those documents in City Hall. That's the nucleus. Ribosomes I think of as a construction company because ribosomes manufacture proteins. So they pretty much build proteins, and proteins are responsible for literally everything in a cell. So they're a construction company, and you wouldn't really be able to have a city without any buildings or anything. So super important. Mitochondria, power plant, or I guess you could say powerhouse. The mitochondria, it produces energy for the cell. So I think that one is kind of self-explanatory because a power plant produces energy for a city. You also have the vacuole, which I think of as like cars or like a delivery service because they pretty much just transport things into and out of the cell and within the cell. So I think that's how I think of a vacuole. So pretty much just like cars that are transporting things. And then you have a lysosome, which is a garbage incinerator. So an incinerator, by the way, is pretty much the place where trash goes to burn. If you guys have seen Toy Story 3, there's an incinerator in that one scene where all the toys are about to get burned. I really like that movie. So lysosome I think of as a garbage incinerator because it takes things that the cell no longer needs and disposes of it. It digests it. It doesn't burn it up, but it digests it. But still, I think garbage incinerator is a really good way to remember lysosome. And then you have the Golgi complex, which I think of as like UPS or FedEx or USPS, pretty much the postal service, right? Golgi complex packages everything and then it sends it off in vesicles. So vesicles, I guess you could say, are the delivery trucks, so like the UPS truck or the FedEx truck. And the Golgi complex is the actual station itself. So you have the people that are packaging up the proteins and then sending it off in the truck, which would be a vesicle. Okay, so we have another question. Which structure is directly responsible for the production of proteins within the cell? So lysosome, Golgi, ribosome, or mitochondria? So I'll give you guys a minute. Okay, so you guys can put it in the chat when you have an answer, but I'll start going through and doing my process of elimination. So production of protein. So I know that the lysosome, which is the garbage incinerator, does not, even if I don't remember exactly what it does, I would know that it does not produce anything. It's responsible for pretty much garbage and getting rid of things that the cell no longer needs. So I would know that the lysosome, which actually it digests things that the cell no longer needs, I know that that doesn't produce proteins. And then I'm going to skip to ribosome. And so then Golgi is FedEx, right? They package things to be exported later. So they don't actually produce anything, right? Even they package up proteins once they have been produced, but they aren't actually responsible for the production of the protein. Mitochondria, powerhouse of the cell, which I say would think of it as a power plant, is responsible for cellular respiration and not at all the production of proteins. Ribosome is responsible for the production of proteins. That is its sole job. It can be on the endoplasmic reticulum, not on the endoplasmic reticulum, wherever it is, it's responsible for producing proteins. Okay, so now we're going to get into cell transport. Remember, if you guys have any questions, please put them in the ask a question box thing, or you can put them in the chat and I'll try to answer them. So we have active and passive transport. So just what do you guys think of? You can put it in the chat when you hear like active. Just like what comes to mind, because stuff like this helps to remember. Don't be scared. So I'll go ahead and tell you what I think of. So when I think of active, I think of like sports or like doing things, going out and running, doing things that are like hard or take a lot of energy. When I think of passive, I think like something that doesn't take a lot of energy. It's not something difficult for you. You just kind of do it and don't really think much of it. Like if you're passively paying attention, you're not actually paying attention to what someone is saying if you're passively paying attention, right? You're just kind of, you hear it, but you're not focusing on it. You're not actually taking the time to think about what they're saying. But if you're actively listening, you're taking notes, you're doing something till you understand what's going on. So also active, again, like athletics, that's what I think of. So that helps in terms of remembering. So the two different types of cell transport, you have active transport and passive transport. So active transport uses energy, right? Just like how I was just saying, athletes do things actively. They use energy to do what they do, right? Play, practice, whatever it is. They use energy. So active transport also uses energy. And the reason why it uses energy is because it moves substances against the concentration gradient. So the concentration gradient of anything, this isn't just biology, but this is just in general, concentration gradient is pretty much the fact that substances like to move from areas of high concentration to areas of low concentration. Like you can think about it logically, right? If you're in a really, really, really crowded room, you want to get out of there. Most people don't like being completely crowded in with other things. You try to get to a little area where it's less crowded, right? So that's pretty much what substances do. They try to get a concentration gradient pretty much is saying that they want to go from more crowded to less crowded. areas. That's it. And then, so moving against the concentration gradient would be moving things from less crowded to more crowded areas, which they don't want to do. So it takes energy to actually do that because it's not what they naturally do. So you have some different types of active transport. These are not all of the types, but these are some specific ones. You have endocytosis, which is pretty much taking in things into the cell, right? Endo usually means in versus exo, which means out or like expelling. So endocytosis is taking things into the cell. So in this picture that I have here, you have phagocytosis and penocytosis. How I learned it was phagocytosis is cell eating, penocytosis is cell drinking. So phagocytosis is pretty much when the cell takes in things that are like bigger, like food particles, solid particles, versus peno, which is the same thing but with liquid particles. So the way that it works, if you look at the image, something touches the cell or comes near it, and something that it wants to take in, and a vesicle is formed as the cell engulfs it in a way, and then it brings it into the cell for whatever it's needed for. And the same thing is true with penocytosis. It's just a liquid instead of a solid. And then exocytosis is pretty much those same processes, just in reverse. So there's not specific ones for like solid and liquid in exocytosis, but it's pretty much just the same thing. It just goes out of the cell instead of coming in. So it goes from the vesicle fuses with the membrane, and then it opens and lets the thing out, or the substance out. So then on to passive transport. Passive transport does not use energy from the cell because it's moving with the concentration gradient. This is already what the substance wants to do, what the molecules in the substance want to do. It doesn't take any extra energy from the cell to make them do this. They go from high concentration to low concentration. And when you just think of it logically, this makes sense because no one wants to be super crowded. So passive transport does not use energy again, and you have diffusion. It's done through diffusion, which can be simple or facilitated. So simple diffusion is pretty much just like it naturally happens. Things diffuse across the cell membrane. They just go from one side to the other without any problems. Remember when I said the cell membrane is selectively permeable? Pretty much that the diffusion, it allows some things to diffuse through. Specifically, the cell membrane allows nonpolar substances to dissolve through because of the whole thing with the hydrophilic heads and the hydrophobic tails. Because it's something polar is hydrophilic. Polar likes water. Polar is soluble in water. So if you have something that's hydrophilic or polar and trying to get through the cell membrane, it'll get stopped because of that whole middle part that is hydrophilic, and it doesn't like water. Versus a non-polar substances like oil, those dissolve, not necessarily oil, but such as oil, dissolve easier through the cell membrane because they can get through that hydrophobic area. Anyway, so that's simple diffusion. Versus facilitated diffusion is pretty much for those bigger substances or substances that are polar that cannot get through on their own, just regular. They need to use proteins like we talked about with the cell membrane. You have transport proteins. Those transport proteins help with facilitated diffusion. So diffusion of water is called osmosis. It's just a specific name for diffusion with water. There's nothing else like special about it other than the fact that it's just with water. So the picture that you have here is of facilitated diffusion. So on the leftmost, you have a channel protein. So that pretty much just provides like a little gap in the cell membrane for things to diffuse through. So like something that's polar or hydrophilic, likes water, can go through that without having to go through the hydrophobic part of the cell membrane, and it's just like a little channel. And then you also have carrier proteins, which actually is the one here that looks almost like a chip clip. Carrier proteins allow large substances to pass through the cell membrane. So large substances that might be too big for the protein channel, or like the protein channel just they don't go together. So carrier proteins allow those bigger substances to go through the cell membrane. And the way that carrier proteins work is, again, all of this is with the concentration gradient. So these substances are still going from high concentration to low concentration. So with the carrier proteins, they just let in the molecule, and then they close, and then they open on the other side. So you might see this described as changing shape, because technically this is a change. It's just pretty much how I think of it is like that, where it just opens and it closes and it lets it out on the other side. Okay, so tonicity is pretty much concentration. So in terms of tonicity, you have hypertonic, hypotonic, and isotonic. So hypertonic pretty much means that there's a higher solute concentration, right? When you think of something that's hyper, or someone that's hyper, it's like a lot. They're just like always doing things. There's a lot. Versus hypo is the opposite. It means that there's less or not a lot. So in terms of tonicity, everything in terms of tonicity is in terms of the solutes. So if you have something that's hypertonic, it means that there are more solutes, which also, in turn, if there are more solutes, there's going to be less water. They're inversely related. But in terms of hyper and hypo, the prefixes are referring to the solutes. So hyper has more solutes versus hypo, which has less solutes. And if it has less solutes, that means in turn it's going to have more water. They're inversely related. And isotonic does not necessarily mean that there is no movement of water. Does not necessarily mean there's no movement of water in general, but it just means that water is moving in and out at equal rates. So there's no net movement of water. So water always goes from hypotonic to hypertonic, because hypotonic means that there is going to be more water versus hypertonic means that there's less water, which can get kind of confusing. So just remember that hyper is more, and it's always in reference to the solute. So be it salt or some ion or something else. Hyper means more solutes, hypo means less solutes. And water likes to go to where there's more solutes. So water always goes from hypo to hyper. So isotonic does not mean that there's not any movement of water, because the concentration is the same on both sides. It really just means that there's no net movement of water. So the water goes in, some goes out, but in the end there's no net movement, which means the amount in and out are equal. So if a cell, for example, which is what you're seeing here in the picture, is placed into a hypertonic solution, that means there's more solutes in the solution. As we said, water always goes from hypo to hyper. So that means that the cell is hypotonic to the solution. Everything has to be in reference to something else. You can't just have a hypertonic solution. It has to be hypertonic in comparison to something else, because it means more solutes. You can't have more than if you don't have a point of comparison, right? So if you place a cell in a hypertonic solution, the water will leave the cell because it's trying to go to the solution, because water, again, goes from hypo to hyper. I'm going to keep repeating that. So when you place a cell in a hypertonic solution, the cell will what's called lyse, pretty much just the cell breaks like a balloon, because all the water is leaving the cell. In an isotonic solution, this doesn't happen because there's no net movement of water. Some goes in, some goes out, but in the end, the cell has the amount of water it's supposed to. And then if you put a cell in a hypotonic solution, which means that there's less solutes in the solution than in the cell, and again, the water moves from hypo to hyper. So the cells are just going to get really inflated, which means that water is going to go in. In this case, the cell lyses because water goes in and it'll like pretty much explode because there's so much water going in. In the other case, I'm sorry, I misspoke, the cell just shrivels up. It does not lyse. Cell lyses when it's put in a hypotonic solution because so much water comes in, versus in a hypertonic solution, the cell simply like wrinkles up because there's no water in it. It deflates like a balloon. Okay, so here we have a practice problem. If you leave your hands in water for a long time, you may notice that your skin swells and begins to wrinkle. This is because your cells are blank to the blank water. The wording of this question can be kind of confusing, so I'll give you guys a minute. You can put your answers in the chat, but the wording does get confusing because you have to have both words in here. So I'll give you guys a minute. Okay, so the answer, I'll go through and again do my thought process. So I know that one of them has to be hyper and one of them has to be hypo, because If it's isotonic, there's not going to be any movement. Your skin is not going to swell. None of that's going to happen if it's isotonic. So that means I can take out answer choice B and answer choice C. So now I'm left with A and D. So one of them is going to be hyper and one of them is going to be hypo, which again, I know gets really confusing with the wording. So your skin swells. If your skin swells, the cells in your skin are swelling. That means that there's net movement of water into the cells, right? So if water is moving into the cells, water again always goes from hypo to hyper. So water is moving into your cells must be hypo. And if water is going into your cells must be hyper, because they move from hypo to hyper, which means the water is hypotonic. I know this is really confusing, and it's just one of those topics. So water goes from hypo to hyper. So if water is going from the bucket or wherever, I'll say a bucket in this case. I'm sorry, it's raining outside. If the water is going from the bucket into your cells, it's going from hypo to hyper. So your cells are the hyper, and the bucket would be the hypo. So the answer in this case would be A. So now I can stop sharing my screen, and we can go ahead and have our Q&A. So please ask me some questions because I'm here to help you and answer them. So you can ask them in the chat if you can't figure out how to use the ask a question box. But if not, I will find some practice questions, I guess, that we can do together. No one has any questions? I know some of that had to be unclear. So please ask questions. Okay, so since we don't have any questions yet, I will go back to the PowerPoint and explain some things that maybe we didn't get to. So in terms of the cell membrane, you also have something called a glycolipid, which is a lipid with a carbohydrate attached. So that's technically not a protein because it's a lipid. So that's just something different. You also have all the different types of membrane proteins. Again, you have transport proteins, receptor, structural, and glyco. And then in terms of the organelles, there are a lot of organelles. So I really do suggest coming up with some type of mnemonic or something, so that way you can know which organelles are responsible for what, because there are so many, it can be hard to just remember them just verbatim, just saying this is this. So mnemonics really tend to help me. So if you guys have any, again, please put them in the chat so we can see, help each other. So in terms of the organelles, again, the central vacuole, which is that huge one in the plant cell, which is the plant cell is on the left, it's the one that's kind of rectangular. So permanent vacuole or central vacuole fill with water, and it helps to give the plant cell some structure. And what's the word for when it's filled with water is called turgid. So you might see that word used. So if it says like the plant cell is turgid, it means that that vacuole is filled with water. So in that case, the opposite of that would be, again, when the plant wilts and the cell would not be turgid anymore because it wilted, which means the water has left that permanent vacuole. So in terms of the hypo and hypertonic, the tonicity that we were just talking about, if you have a plant cell in a hypotonic solution, the cell would become turgid. Turgid is the word. So because, again, water goes from hypo to hyper. So if you put the plant cell in water, the water's in a hypotonic solution, the water wants to go from hypo to hyper, so the water will move into the plant cell, and this permanent vacuole will fill, and it'll be called turgid. Versus if you put it in a hypertonic solution, the water will exit the permanent vacuole, and the plant will wilt, I guess, because it'll like wither or wrinkle because it doesn't have enough water inside of it. We also have mitochondria and chloroplasts. So plants have both of them, which is a common misconception. They do not just have chloroplasts, they actually have both because they still need to produce energy. What chloroplasts do is they take energy from the sun and they turn it into sugar, but they don't turn it into energy. So the plant still would not have any energy to do any of its cellular processes. So it needs mitochondria because the mitochondria then takes that sugar and turns it into energy. So in us, we don't have chloroplasts simply because we don't conduct photosynthesis. We eat to get our sugar. So that's how we get it, which is why we don't need chloroplasts. But plants don't eat most of them, so they need chloroplasts and mitochondria, not just chloroplasts. So also in this diagram, you have what's called the nucleolus, which is pretty much just the center of or an area of the nucleus that's even more, that has a lot of the DNA in it. You have microtubules and filaments and all of that. They just help to give the animal cell structure because the plant cell has a cell wall for that. Animal cell doesn't, so filaments and microtubules and all of those help with that, and they also help with cell division. Peroxisome is pretty much like a lysosome. It just helps with it digests things using hydrogen peroxide. Let's see. So again, back to the mnemonic, because I think that it's very helpful for me. I've also seen people do the cell as, what have I seen? I've seen the city, I've seen like a factory or like a business. And I think I've also seen the human body, but the human body one I'm not familiar with like that much. I've seen it used though. So whatever helps or works for you. If you have something different, that's totally fine. It's also, as long as it helps you remember. So you have the cell membrane as the city limits again. You have cell wall, which is again only in plants or in prokaryotes like bacteria. That is the fortifications, right? Like the wall, the moat, the whatever you want to call it, of a city, or I guess we don't really have many fortifications, so I guess it would be like a town or an ancient city or something. You have the nucleus, which is City Hall, because it stores all of the information. So that's where a whole bunch of files in City Hall, the information on everyone is in City Hall, everyone who lives in the city, that's all found in the nucleus. And then you have ribosomes, which I think of as construction company, again, because they pretty much build proteins. They are responsible for building proteins. You have mitochondria, which is the power plant, not necessarily the powerhouse, but the power plant, because it produces energy for the cell. So power plant in a city produces power for the city, which is in the form of energy, you know, lights to power, electricity to power your lights, and all of that. So mitochondria produces energy to power cellular functions. And then you have the vacuole, which is pretty much cars or delivery. So if you want to think of it in terms of the Golgi being UPS or FedEx, the vacuole would be their trucks. So the vacuole is what delivers things, the proteins, different places. And then you have lysosome, which again would be the garbage incinerator. It takes things that the cell no longer needs, which can be old cell parts. It can be any type that they're not using anymore. It can be any type of waste from the cell or any type of bacteria or microorganisms that could get inside the cell. Lysosome takes that and it digests it. It has digestive enzymes and it digests. You also have the your Golgi complex / Golgi apparatus. Same thing, just different names for the same thing. Those are, I like to think of as UPS or FedEx because they package things. So like the actual UPS store packages things and gets ready and sends it off in the truck. The truck is the vacuole. The truck is the vesicle. And then Let's see. I know I've also seen the cell as a factory, so that might help some of you guys to remember better. So I don't know all of them for the factory though. So if you have the cell membrane would be like the limits again of the factory. The cell wall would be some type of wall around the factory. I mean, the cell wall is kind of self-explanatory in terms of these. The, I guess, center of the factory, like production center, would be the nucleus. Ribosomes would be the construction company. Ribosomes would be the actual like workers in the factory because they're the ones who are doing things and like producing whatever the product is at the factory. But in a cell, they produce proteins. Mitochondria would be the factory still needs power, so I guess it would be the power plant. The vesicles would be like, you know how on a factory they have what's the word? The things that move the product from place to place. In a factory, that would be in a cell, though, that's the conveyor belt. In a cell, that's the vesicle. Lysosome, I guess, would just be the trash. And then Golgi complex would be the actual packaging center of the factory. So back to cell transport. You have active transport and passive transport again. Active transport moves it against the concentration gradient. So that's not, the idea of concentration gradient is not even unique to biology. That's something you also learn in chem and other sciences. It simply says that substances, be it atoms, molecules, ions, whatever it is, like to move from places of high concentration to places of low concentration. So when you need to make them do what they don't want to do, that takes energy. That's active transport because they're moving against the concentration gradient. And then passive transport, on the other hand, they're already doing what they want to do ideally, so you don't need to use energy to make them do what they're doing already. Active transport takes energy because you're making them do something that they do not usually do, that they do not want to do in the first place. So in terms of active transport, you do have endo and exocytosis. And in endocytosis, you have phago and pinocytosis. And you also have something called receptor-mediated endocytosis, which pretty much means, which is very similar to phago and pino. It's just that there's a receptor on the cell that hits, that the substance hits the receptor, and then the vacuole forms and engulfs it. And usually that's a little bit more complicated. It involves some other processes that'll happen later in the cell, depending on what the substance is. And then in terms of passive transport, does not use energy. You have osmosis, which is diffusion of water, and there are simple and facilitated diffusion. Simple diffusion in terms of the cell is usually for nonpolar small molecules because they can pass through the cell membrane without any problems, versus facilitated diffusion, which is when the molecule is polar or large, just kind of too big to fit through the cell, to just float through the cell membrane like the other ones. So with simple diffusion, you have the protein, you can have protein channels like you see in the leftmost of that image. You can have carrier proteins. But either way, it's going from high concentration to low concentration, which is what it wants to do anyway. So it does not take any extra energy to actually do this, even if it's using proteins. And then back to tonicity, you have hypertonic, hypotonic, isotonic. Water always goes from hypotonic to hypertonic because, again, the hypo and the hyper are referring to the amount of solute, which is the thing that's dissolved in the water. So if it's salt or some sort of ion or some other molecule, water is going to go to where there's more of that molecule because that in turn means that there's less water. They have an inverse relationship. So just to clarify what I said earlier, if it's hypertonic, that means you have a higher solute concentration. And in that case, the water will all exit the cell and the cell will almost like wither because it doesn't have enough water in it. Versus because water moves from hypotonic to hypertonic. And the solution is hypertonic in relation to the cell. Isotonic means that they both have the same concentration. So water moves in, water moves out, but in the end, the same amount of water is still in the cell. And then you have hypotonic, which means that the cell is going to lyse, which means it's going to explode pretty much. because water moves from hypotonic to hypertonic. So water is going to go into the cell, and pretty much the cell is going to fill too much with water, and it's going to burst kind of like a balloon. So again, the hyper and the hypo are in reference to the solute concentration, not necessarily to the water. It's reversed when it comes to the water, because more solute means less water. And more water means less solute. So that can be kind of confusing. So that, I would say, is something that you would really have to look into a lot, because of just the way it's worded and how you have hyper and hypo, and how they're kind of backwards for the water. But water always moves from hypotonic solutions to hypertonic solutions. So water moves from places with less solute to places with more solute, to kind of dissolve it more, I guess. And then I'll go back through the multiple choice that we did in case any of you guys missed it. So if you leave your hands in water for a long time, you may notice that your skin swells and begins to wrinkle. This is because your cells are blank to the blank water. So my thought process, again, you know that one of them is going to be hyper and one is going to be hypo. If it's isotonic, your hand wouldn't swell, so that you can take out B and C, right? And then, again, you have to remember water goes from places that's hypotonic to places that are hypertonic. So if waters are moving into your cell must be hypertonic. So that would tell you that the answer would be A. And if your cells are hypertonic, that means the solution is hypotonic. One is always going to be one of them, the other one's going to be the other one. So you should have been able to narrow it down to A and D. And then I'll go, again, if you have any questions, please you can just put them in the chat, and I'll look at them and answer, because we only have a few minutes left. So which structure is directly responsible for the production of proteins within a cell? So again, my thought process here is I do process of elimination just to be sure that there's no other possible answer, because you never know. To double check that your initial thought is right. So my initial thought is going to be C, ribosome, which is the answer. But in terms of process of elimination, if I couldn't remember what a ribosome does, ribosomes produce proteins, that is their function. But if I couldn't remember that, I would go through and use process of elimination. You have lysosome. I know lysosome is the incinerator. Lysosome digests pretty much garbage in the cell waste. So I know that's not responsible for production of proteins. Golgi apparatus, I know, is involved in proteins, but not the production. They just package them and send them off. I know that, so that's not responsible for the production of proteins. They're responsible for the packaging and export of proteins. And then mitochondria, I know, is unrelated to proteins. It's responsible for it's a powerhouse, responsible for cellular energy and producing energy in the form of ATP in the cell. So I would use process of elimination there. And then here, you have which of the following best characterizes the structure of the plasma membrane. You have rigid and unchanging, rigid but varying between cells, fluid and disorganized, and fluid and organized. So in this case, the answer would be D, fluid and organized, because again, the cell membrane or plasma membrane is the same thing, or phospholipid bilayer, same thing. That's referring to the fluid mosaic model. So fluid, obviously, and given in the name fluid mosaic model, is going to tell you that it's not A or B. So you have it down to C and D. And it's going to be D because it is organized. It's not disorganized. Even though they're all floating and kind of moving, it's still organized. Each thing still has a specific function and specific place within the cell membrane. And then we can go back to the first question. So studying a picture of a cell taken with an electron microscope, you find that the cell has no nucleus and no mitochondria, but it does have a plasma membrane and a cell wall. You conclude that the cell is probably from A. So you can take out D because that's just kind of irrelevant. That has nothing to do with the question. Like the type, in terms of types of cells, extinct organisms have the same type of cells as far as we know. So that answer choice is kind of off the table. And then you have animal, plant, and prokaryote. So animal and plants are both eukaryotes. So if the cell has no nucleus and no mitochondria, it's not an animal or a plant because it's not a eukaryote. They both fall under that umbrella term, which tells you that it's C, a prokaryote. Because again, prokaryotes have no nucleus, no mitochondria, no organelles. So they don't have nucleus, they don't have mitochondria, they don't have chloroplasts. If it was a plant, they don't have organelles. So here's the picture of the prokaryote. So they do have ribosomes, they do have cytoplasm, they do have a cell wall, but they do not have any organelles. So ribosomes produce proteins. I guess that's one thing that you would consider like closest to an organelle, but it's not necessarily. And that's how you would be able to tell prokaryotic from eukaryotic. So that's all I have for you today. Thank you for attending, and that's it. Thank you. I hope you learned a lot. Let me close this.